Imagine a bustling factory, working tirelessly to create everything our bodies need. This is the world inside our cells, where microscopic machines called ribosomes hum with activity. These remarkable structures are responsible for one of life's most fundamental processes: protein synthesis. Without ribosomes, our cells would grind to a halt, unable to perform even the simplest functions. At the heart of these molecular machines lies a fascinating molecule called ribosomal RNA, or rRNA. RNA is a crucial component of ribosomes, playing a central role in reading genetic instructions and assembling proteins. Understanding rRNA unlocks the secrets of how our cells function and provides a glimpse into the very essence of life itself. To understand the importance of rRNA, we must first delve into the fascinating world of RNA and its various functions. RNA, or ribonucleic acid, is a crucial molecule that acts as a messenger, carrying genetic information from DNA to the ribosomes, the protein factories of the cell. DNA serves as the blueprint of life, containing all the instructions our cells need to function and thrive. However, DNA resides within the cell's nucleus, a protected compartment that safeguards this precious genetic material. RNA acts as a courier, delivering these vital instructions to the ribosomes in the cytoplasm, where proteins are synthesized and assembled. There are different types of RNA, each with a specific and essential role in the process of protein synthesis. Messenger RNA, or mRNA, carries the genetic code from DNA to the ribosomes, ensuring that the correct proteins are made. Transfer RNA, or tRNA, brings the building blocks of proteins called amino acids to the ribosomes, facilitating the assembly of proteins. And then, there's rRNA, the star of our show, which forms the structural and functional core of the ribosomes themselves, playing a pivotal role in translating genetic information into functional proteins. Ribosomes are the protein synthesis powerhouses of the cell. They are complex molecular machines found in the cytoplasm of all living cells, from the simplest bacteria to the most complex animals. Ribosomes are composed of two subunits, a large subunit and a small subunit, which come together like puzzle pieces to form a functional ribosome. Each subunit is made up of both rRNA molecules and proteins. The rRNA molecules provide the structural framework for the ribosome, while the proteins help to fine-tune its function. Think of the rRNA as the scaffold of a building, providing the basic shape and support, while the proteins are like the walls, windows, and wiring that make the building functional. Ribosomal RNA is a remarkable molecule. It is not only a structural component of the ribosome but also plays a critical role in its function. rRNA helps to position the mRNA and tRNA molecules correctly within the ribosome, ensuring that the genetic code is read accurately. It also catalyzes the formation of peptide bonds, the chemical links that hold amino acids together to form proteins. In essence, rRNA acts as a molecular matchmaker, bringing together the mRNA with its genetic instructions and the tRNA carrying the corresponding amino acid. This intricate dance of molecules ensures that proteins are synthesized correctly, building block by building block, according to the instructions encoded within our DNA. Section 5. The Dance of the Subunits, How Ribosomes Assemble. The assembly of ribosomes is a fascinating and intricate process that highlights the dynamic nature of these essential molecular machines. The rRNA molecules for both the large and small subunits are transcribed from DNA in the nucleolus, a specialized region within the cell nucleus. This transcription process is highly regulated to ensure accuracy. Once transcribed, the rRNA molecules combine with ribosomal proteins to form the individual subunits. These proteins are imported into the nucleolus from the cytoplasm, where they meet the rRNA. These subunits then exit the nucleus and enter the cytoplasm, where they remain separate until protein synthesis is needed. This migration is a critical step in the assembly process. When a cell needs to make a protein, the small and large ribosomal subunits come together, sandwiching an mRNA molecule between them. This union is essential for the next phase. This triggers the process of translation, where the genetic code carried by the mRNA is deciphered and used to assemble a specific protein. The ribosome reads the mRNA sequence and translates it into a chain of amino acids, forming a new protein. Section 6. A tale of two ribosomes, prokaryotes versus eukaryotes. Ribosomes are found in all living organisms, but there are some subtle differences between the ribosomes of prokaryotes, bacteria and archaea, and eukaryotes, plants, animals, fungi. Prokaryotic ribosomes are slightly smaller and less complex than eukaryotic ribosomes. Prokaryotic ribosomes are composed of three types of RNA, 5S, 16S, and 23S. Eukaryotic ribosomes, on the other hand, contain four types of rRNA, 5S, 5.8S, 18S, and 28S. The S refers to Svedberg units, a measure of sedimentation rate, which reflects the size and shape of the rRNA molecules. Despite these differences, the basic function of ribosomes, protein synthesis, is conserved across all life forms. Section 7. The Rhythm of Translation. RNA in action. This intricate dance is fundamental to life itself. The process of translation, where the genetic code is translated into a protein, is a marvel of molecular choreography. It's a highly coordinated event that ensures the accurate synthesis of proteins. The mRNA molecule, carrying the genetic instructions from the DNA, threads through the ribosome like a tape measure, guiding the process with precision. Each three-letter code on the mRNA, called a codon, specifies a particular amino acid. These codons are the language of the genetic code. The rRNA within the ribosome helps to position the mRNA and tRNA molecules correctly, ensuring that the codons are read accurately. This precise positioning is crucial for the fidelity of protein synthesis. The tRNA molecules, each carrying a specific amino acid, bind to the mRNA codons via their anticodons, three nucleotide sequences that are complementary to the codons. This binding is essential for the correct assembly of the protein. As the ribosome moves along the mRNA, it links the amino acids brought by the tRNA molecules together, forming a growing polypeptide chain. This chain will eventually fold into a functional protein, performing vital roles within the cell. Ribosomal RNA is not only essential for life but also serves as a powerful tool for understanding evolutionary relationships between organisms. Because rRNA is so fundamental to cell function, it has been highly conserved throughout evolution. This means that the RNA sequences of closely related organisms are very similar, while the rRNA sequences of more distantly related organisms show greater differences. By comparing the rRNA sequences of different organisms, scientists can construct phylogenetic trees that depict the evolutionary history of life on Earth. rRNA analysis has been instrumental in our understanding of the early evolution of life and the relationships between the three domains of life: bacteria, archaea, and eukarya. Ribosomal RNA is an indispensable molecule for all living organisms. Without rRNA, protein synthesis would grind to a halt, and life as we know it would cease to exist. RNA plays a central role in translating the genetic information encoded within our DNA into the proteins that carry out all the functions of life. From the enzymes that catalyze biochemical reactions to the structural proteins that give our cells shape and support, proteins are the workhorses of the cell. And rRNA, the master architect of the ribosome, ensures that these proteins are synthesized accurately and efficiently, enabling life to flourish in all its diversity. While rRNA is best known for its role in protein synthesis, recent research has revealed that its functions extend far beyond the ribosome. Scientists are discovering new and exciting roles for RNA in gene regulation, cellular stress responses, and even viral infection. These discoveries underscore the complexity and versatility of RNA molecules and highlight the importance of continued exploration into the hidden world within our cells. rRNA, once thought of as a simple structural component of the ribosome, is now emerging as a key player in a wide range of cellular processes, opening up new avenues for scientific inquiry and potential medical applications. The story of rRNA is a testament to the elegant simplicity and astonishing complexity of the natural world. From a single molecule, a universe of activity unfolds within our cells, driving the processes that make life possible. rRNA, the unsung hero of protein synthesis, reminds us that even the smallest components of our cells can hold the keys to understanding the grand scheme of life. As we delve deeper into the mysteries of RNA, we are constantly amazed by its versatility and its central role in the grand symphony of life. The exploration of rRNA has only just begun, and the future promises even more exciting discoveries that will illuminate the intricate workings of our cells and the very essence of what it means to be alive.